Okay, ecology class, welcome to this online lecture for some of the material in Chapter 8 talking about populations, abundance, and distribution. And make sure that you're also paying attention to your emails while I'm gone, and we'll try to check in with each other that way as well. So in this lecture, I'm going to talk about population abundance and distribution from the perspective of practices, meaning how do we measure the sizes of populations, how do we look at distribution patterns, and so then we'll look at those patterns that we see in terms of how organisms distribute themselves in the environment, and then try to determine what processes may be responsible for those patterns. So let's begin with a reminder of how we defined population to begin with. Remember that a population is a group of individuals of the same species, right? And the fact that they live in a particular area where they can interact with each other, including mating, so they reproduce mostly with one another within that population, is an important part of the definition. And remember that populations can be really small and they can be really enormous, and that often the population is defined by the investigator. When we talk about population distribution, we're talking about how they're distributed over a geographic area. And we've already talked a little bit about this before when we were talking about ranges, and we were talking about fundamental and realized niche previously. Abundance is obviously just the number of individuals. And so some terminology for you is that we'll often see used, and sometimes people confuse them a little bit, which is one of the reasons why I want to just clarify it here, is that population size means the abundance, the total number of individuals, right? Whereas population density, which is another term that we'll see, is more of a relative measure of abundance. So a density is a number of individuals per unit area, or in the case of aquatic organisms, maybe the number of individuals per unit volume. So we can have number of deer per hectare, or number of starfish per square meter. And we're going to talk about these examples in just a minute here. So what we're talking about then is the idea of measuring absolute abundance versus relative abundance. So we can then move on to looking at methodology. How can we count or estimate the total number of individuals in the population? That's the absolute abundance. Or how can we get an idea of the relative number of individuals per unit area? And that's our, of course, our measure of relative abundance. One thing we can do is to do timed samples, where we might count the number of these little sea urchins along the shoreline and try to see how many we can count in an hour, or in 2 hours, or in 5 hours in particular areas. We can sometimes use evidence of organisms in their environments by looking at things like footprints. So there's a variety of different ways, and we're going to go over some of these methods for how we can estimate population size and density. One common way that people estimate absolute abundance is to use something that are called quadrats. And quadrats are usually like these small squares, although a quadrat can actually be circular as well. But it's a defined area. So you would choose a quadrat, say 1 meter by 1 meter on each side, and then you know how many animals you have in a square meter. Now, of course, the best way to kind of sample would be to count every single organism in your area. And this picture here shows if you had some kind of a isolated tropical island, maybe you could actually go in there and count every single sea urchin, or every single insect, or every single tree. Maybe wouldn't be so hard on this little island. But usually, because it's impossible to count the total number of individuals in the area, we use a sampling method. We talked about sampling when we were talking about the arthropod lab. We couldn't count the total number of arthropods in the forest versus the field, and so we did sampling by taking sweep samples. And the Quadrat method is just another way of sampling. So there's different ways of placing your quadrats around. You can place quadrats at random points within a larger area. You can place them along a line that we will call a transect line. Okay, and there's different ways of utilizing a transect line, but one way would be to place your quadrats evenly spaced along a transect line. Here's just another photograph of a plant ecologist using a 50 by 50 centimeter quadrat here in order to count the number of plants for every 0.5 meters squared. Okay, so how do we go from counting numbers of individuals within a quadrat to estimating our total population size? So of course it helps to know at least what the total area is that you may be working under. If you have a large field here that is, say, 100 meters by 100 meters, and so your area is going to be 10,000 square meters, okay, and then you are able to place a number of quadrats. Let's say that we are trying to estimate the size of a plant population, because this quadrat method is certainly going to work better for organisms that are sessile, ones that hang out there and don't move around much, as opposed to organisms that are mobile and do move around. Okay, so the quadrat sampling method is definitely best for sessile organisms, or sedentary organisms is another name for that. So if this is a 100 meter by 100 meter plot, we know we have 10,000 square meters altogether, and that if we were to place a number of quadrats in here in order to count our number of plants per square meter, and let's say we're using a 1 meter by 1 meter quadrat, well, first of all, do we want to just place one quadrat and then we get 5, and so we say we have 5 plants per square meter? Or do we want to do what? No, we want to do several quadrats so that we have several samples, right? And then what are you going to do with those numbers? Let's say we take 10 of them, or 15, or 20. What do we want to do? We want to take the mean, or the average, right? So that we end up with the average number of individuals per square meter based on a sample of, say, 20 quadrats. All right, and so once we have that figure, okay, so let's say we have an average number after taking several quadrats, and we end up saying we've got an average of 8 plants per square meter from doing that kind of sampling. Then how do we go from our relative measure of density to an estimate of total population size? Well, if we have 8 plants per square meter and our total area is 10,000 square meters, then how many plants do we have? Okay, we're just going to multiply 8 times 10,000, so we're going to have a plant population size of 80,000 individuals. Right? It's pretty straightforward. And if you have closely spaced plants like this, it may be that you have quite a few. One of the things that you're probably thinking about already, and this is an idea that we're going to come back to in a little bit, is how the plants are arranged in this area. If they're all clumped together, or if they're uniformly distributed, or if they're spread out in different ways, that's going to affect our quadrat sampling method, right? So quadrat sampling is definitely going to be influenced by the distribution of organisms within the total area. And again, we're going to come back to those dispersion pattern measurements in a little bit. So another fun way to try to estimate population size for organisms, especially those organisms that are mobile, is to use something called this mark recapture technique. And you guys are going to get a chance to use the mark recapture technique in class next Tuesday while I'm gone. It's going to be the lab that you're going to do. And unfortunately, you're not going to be able to do it with Rocky Mountain goats, but you are going to do a simulation with some beans. And you'll find out more about that in class. But the way that this technique works is that you have an organism, usually a mobile organism, as we talked about before, those that kind of move around a lot. And then you're going to mark some individuals, okay? And then what you're going to do is capture a sample. So let's say we go out and we find a number of mountain goats. We can trap them some way or capture them in some way, and we mark some of the individuals. We can't mark all of them. If we could mark all of them, we could just count them. So we mark a small sample, and then what we do is we allow the marked individuals to go back into the population and then to randomly mix with the other individuals that are there. And then we take a second capture, a recapture, right? So we go out and we capture a bunch of new mountain goats, and so some are going to be marked and some are not going to be marked. All right, let's look at the next slide here for a minute.